Today topic of discussion is prion. Prions are even more primitive than virus because prion have only protein. Virus it have protein plus nucleic acid, either DNA or RNA. So only protein means prion, along with the nucleic acid means virus. Prions are infectious that are causing diseases. Let's see how they work. Prion are infectious protein particles without nucleic acid. This one is the infectious protein particles without nucleic acid. They have two isomers. One is PrPC, another one is PrPSc. Prion protein Scrapiee. This one is disease causing form of prion protein. This one is the disease causing form of prion protein. This one is just protein. It is found in our body. This one, why this one having Scrapiee? Why? Because means the sheep always scratches itself because it was so itching. Scratch itself everywhere. So it is a infectious one. PrPSc, it carry itself to neuron and it is carry to neuron to this PrPSc. This PrPSc bind with PrPC. PrPC. It is a cellular protein found in our body. So this to bind, it will converted into PrPSc. Now so many PrPSc, they get aggregated. They causes amyloid like plaques. So these are some insoluble protein. Amyloid insoluble protein. So amyloid like some plaque are created. Basically a lack of immune response. Slow virus under this heading it coming. The body will not get immune response against this. There will be lack of inflammation also. And now what happened? The cells internalized by neuron itself. Internalization. The neuron internally taking by PrPSc. That neuron, this one is the neuron means it internally taking that PrPSc. The PrPSc are accumulated by the cytoplasmic vacuoles, cytoplasmic vacuoles here accumulated, giving the cell spongiform appearance. So it giving cell to spongiform appearance. All the vacuoles containing what? All the vacuoles containing PrPSc. PrPSc bind with PrPC, where it is going to nerves. The nerves internally takes place taking up this PrPSc. They accumulated into the cytoplasmic vacuoles, giving the cells like spongiform appearance. The cell look spongiform. The vacuoles containing PrPSc. That's why this is called as spongiform encephalopathy. The neuron are affected. So what is causes of prions? Basically what and all we hear: vacuolation of neurons, plaque will be there, proliferation of hypertrophy of atrophy is there, then fusion of neuron and adjacent glial cells. What are the causes? Vacuolation, plaque, then hypertrophy, astrocytes, then fusion of neuron, adjacent glial cells. Let's discuss slow viral diseases due to prion. First one is in human. First disease Kuru. Kuru means drama. This was first identified in 1957 in tribe in New Guinea. This one incubation period 5 to 10 years. Infection believed to be due to cannibalism. Okay, what is cannibalism? This is the tribal custom of eating dead bodies. Okay, eating dead bodies of relative after ritual non-sterilizing cooking. After ritual non-sterilizing cooking. The abolition of cannibalism, the disease has disappeared. Second disease name is called as Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease, CJD. This one is the most common in human. Dementia, myoclonus and death. So CJD causes dementia, myoclonus and death. This may be familial and aerogenic. Then what are the sources for CJD? This can be occurred by improper sterilization depth electrodes or cornea from infected persons transplanted to other person, in human growth hormones given from infected persons, the durameter graft and gonadotropin which is received from infected person, then through surgical instrument. So CJD is also known as iatrogenic disease when physician induced this disease. Dementia, myoclonus death, then aerogenic infection from physician, it induced the disease. This one also familial. Gerstmann Schwarzer Skinker disease, GSSD, present with a slowly progressive limb and trinical ataxia as well as dementia. This one predominantly involved involvement of brainstem degeneration. The neuropathology of GSS is remarkable in that extensive and invariable amyloid deposition occur. Next one is fatal familial insomnia. What are the presentation? Insomnia, dementia, motor paralysis, then hyperthermia, hypertension. These are the things. So what are the human diseases? First one is Kuru, second one is CJD, third one is GSSD, fourth one is fatal familial insomnia. In animals, Scrapiee, sheep, goat, mice and mink. Bovine Spongiform encephalopathy. The host is cattle. Transmissible mink encephalopathy. Host mink. Chronic wasting disease. This one host mule deer and elk. It is a prototype of Prion disease. There are the two types, natural Scrapiee and experimental Scrapiee. This one, natural Scrapiee, that incubation period of 2 years, affected sheep become irritable. So incubation period 2 years, affected sheep become irritable. It have intense to very itching for it. So basically what happened in the sheep, after the incubation period of 2 years, they vertically from parent to offspring it can come or direct contact the sheep got it. 2 years of incubation period, the affected sheep become irritable and it develop intense scraping themselves against trees and rocks. So the name given Scrapiee. Trees and rock scrap their body. Finally, they causes paralysis and death. Second one is, what is that experimental Scrapiee? This disease can be experimentally transmitted to various animals. So they are injecting the neural tissue of infected sheep to the other animals like hamster, mice. So they are getting from infected sheep to study the progress of disease, etc. Mad cow disease caused by these prions. Foot and mouth disease caused by virus. Mad cow disease caused by prion.